your lecture. It's 6 o'clock now. Are you okay if we started it?
Yes, yes,
I
sure.
think there are some some people who would probably join, but I think I'll kind of not wait. Rather, I'll give the audience a introduction about you and Anindo, and then we will start. So, good evening
Sure.
and welcome to this InSIS webinar series 2023. So, this is probably the last webinar organized by InSIS and IIT Madras on this year 2023. And we have with us the distinguished speakers, Professor Kaushik Chatterjee from the Materials Engineering Department of Indian Institute of Science, and Dr. Anindo Roy, who is a postdoctoral research associate from Department of Materials Engineering, IISc, Bangalore. So they will talk about the nanomechanics of bacterial cell wall damage via high aspect ratio bioinspired nanostructures. So it's kind of, in the higher uh, domains that we are looking at this topic compared to the conventional fatigue and fracture that most of the members are aware of. And to give a brief background about the speakers, Professor Kaushik Chatterjee is received his basic degree from Bengal Engineering College, then MS and MS from the University of Virginia, and PhD from Penn State. He worked as a postdoctoral fellow, jointly at the NIST and National Institute of Health. He joined IISc Bangalore in 2011 and is currently a professor in the departments of Material Engineering and Bioengineering. He is also the chair of the Department of Bioengineering. His group works on materials for biomedical applications, and in the recent years is particularly focused on the use of 3D printing and additive manufacturing technologies for healthcare. He is currently an associate editor for the Journal of Materials Chemistry B and Materials Advances. Next is Dr. Anindo Roy. He completed his bachelor's from the IIEST, and that's in Shibpur. And he then joined the IISc as a graduate student, and his doctoral work focused on generating bioinspired nanoscale architectures on clinically and biomedically relevant surfaces for the development of new age chemical-free antibacterial surfaces. He is working currently as a research associate at the Department of Materials Engineering, IISc, and his research interests include bacterial traps, functional surfaces, and bacteria nanostructure interactions. So with this few words of introduction, I will now request Professor Kaushik Chatterjee and Dr. Anindo Roy to deliver their lectures.
Thank you. Thank you to, first of all, InSIS as a society for the kind invitation, and also to Professor Raghu Prakash, Professor Vikram Jairam, and others. Uh, as well, some of you know me, but for others, I mean, I have degrees in which for every degree there's a different name, but nothing that qualifies me to talk about structural integrity. This is not my forte or my focus area. Having said that, we do look at materials for biomedical applications, and when you do that, when it comes to biology or healthcare, mechanics at multiple length scales play a very important role. From matching the mechanical compatibility of materials to various tissues, and all the way down from the nanoscale to the macro scale. And here, I want to particularly talk about some work that's been ongoing in our group for more than a decade now. We've been looking at topographies to kill bacterial cells uh, and use essentially mechanical or physical forces to damage bacteria, in contrast to more conventional strategies which use chemistry or biomolecule-based approaches to tackle the challenges associated with bacterial colonization of surfaces and also bacterial infections. So, and with me is Anindo. He has actually been working on this for the last 6 years for his PhD. It's earlier this month that he officially defended it, and which I thought qualifies him to be in the line of fire today and basically explain the details of what he's understood about the nanomechanics and also some of the work that he's done. So what we plan to do is basically start with a general introduction of all of this, and then I will, after the first a few slides, I will hand it over to Anindo. He can explain much better about some of the current models of what is known about these mechanisms or the of these mechanics at that nanoscale between surfaces and bacteria, and then some new ideas that we've been trying to develop in our own lab, and how that will compare and contrast with things that are already reported in the literature. So Anindo, can we move to the next slide? Yes. This is just more of a broad overview of this I would say the challenge, the societal challenge that people often encounter when it comes to bacteria, and our everyday lives where bacteria are essentially everywhere. They're inside us. They're the good bacteria, the probiotic ones, if you will, and then there are many others which we wish weren't there, but we do deal with them all the time. So when it comes to the bacteria coming in contact with any surface, what is typically reported is that, or understood, is that they start off in the planktonic state, which is in single suspensions. So this is what you see here on the left, with these single yellow cells that are coming in contact with the bacteria, I mean, with the surface, and then they start to adhere. So initially, it's more of a physical adsorption. Sometimes there are some chemical bonds formed, but then what the bacteria do is start secreting a lot of biomolecules, and then form what is go on to form a colony, and then eventually like a what's called a biofilm, where there is a mix of cells with a lot of extracellular proteins which they secrete, and that gives rise to a very, uh, I would say, challenging state where it's difficult to treat them with antibiotics. So, a lot of the effort in the field is to try and see if you can kill the bacteria when they're still in single cell form and not able to form the biofilm. And within this biofilm, of course, the other difficulty is that once it's formed, it's difficult for antibiotics or other chemicals to penetrate this mass of cells and proteinaceous mix. And therefore, it's all the more reason to try and prevent this. And a lot of effort has gone into developing surfaces that can resist the attachment of bacteria. And I'm going to try to tell you about how the new thought is that you can kill them instead of just trying to resist them. In terms of why this is important, there are various fields or various aspects of our everyday life where this becomes important. One is when you have implants going inside the body. So you could have anything from dental implants, orthopedic ones. It could even be soft materials like your contact lenses. Any of them could get affected when bacteria would colonize the surface, and then they form the biofilm on it. And also, what I forgot to mention here is the biofilm becomes a source of the bacterial cells starting to spread into secondary locations. So you want to minimize that, and you can see this image on the right, where there's a retrieved femoral head of a hip implant, where there's a bacterial biofilm that is essentially formed on this surface. The next situation is the hospital-acquired infections. So you could have a patient who already is infected, and they will I mean, because maybe they're coughing or they're touching a surface, that they transmit that bacteria onto another surface in the hospital. So whether it's the caregivers or anybody else, other patients, they can acquire that bacterial infection either because they also touch it, or then there is the contaminated equipment and other sources by which it gets transmitted, and that becomes a major challenge in trying to maintain a clean environment in the hospital. Other areas which don't necessarily deal with healthcare but are important include packaging for food, where the food contamination becomes a challenge. And then there's also other areas like, say, pipelines, where bacterial colonization can damage by corrosion or other chemical actions from what the bacteria would secrete. So, Anindo, the next slide. So the other, sort of challenge that has come up in particular for healthcare is that these bacteria have become increasingly more resistant to antibiotics, and this is known as antimicrobial resistance, or AMR, and it's widely recognized by World Health Organization and others as one of the gravest challenges of healthcare, probably more so than even COVID and other things, because the fear is that in another 50 years or so, we were going to run out of all kinds of antibiotics, and all the uh, the developments or the advantages that we've had in terms of modern medicine being able to tackle infections will essentially be lost, because even the rate at which we are now developing antibiotics is not keeping up with the rate at which we are essentially generating new strains that can resist them. So this is a real threat, and you can see here that these numbers are quite grave. People are talking about easily, 10 million deaths by 2050, which would be quite catastrophic. Also, there's a lot of work in the molecular and the microbiology field trying to understand what happens. So essentially, what the understanding is that these cells can develop the resistance to these antibiotics through various mechanisms. One is by coming up with new molecular pumps that can push the antibiotic out, so that the concentration inside the cell will become much lower. They also have other mutations, so that they can change the nature of their cell wall, and also new enzymes that can essentially degrade these. And so as a result, these increasingly new strains can resist uh, antibiotics. And in fact, even what are called the last line, or the really sort of toxic doses of antibiotics have to be administered to try and tackle bacterial infections in many patients. So on into the next one. Right. So in nature, there are various surfaces where people have observed that these are topographies that would be that do impart a variety of benefits to the materials, and that has been of much interest for several decades now. And you could probably categorize them into these few subgroups based on the length scales at which these topographies are seen. So one is, of course, these the roughness, where it is on the length scale of a few microns. And here, for comparison, the bacteria is about 1 to 2 microns. Then there are length scales where they are about just at the size of a bacteria, so maybe about a micron. And then there are newer surfaces or newly discovered surfaces where the length scale is a few hundred nanometers, such that they are much smaller than that of the bacterial cell. So this has many implications. Number one is, of course, people have been looking at these surfaces for things like, generating hydrophobic surfaces, also self-cleaning. In the West, there's a lot of interest in making surfaces for that can, for de-icing. There's also now interest in trying to use them for collecting water in very arid areas. But in the case of healthcare, and particularly bacterial infections, people are trying to see whether one could use these kind of surfaces to try and resist the colonization of bacteria. So when you think about lotus leaves, rose petals, you're talking about typically topographies that would lie in these first two categories on the left, which would be on the micro scale. So what happens here is the bacteria can, when it comes in contact with these kind of surfaces, in contrast to the flat ones, they're unable to decide where to sit, whether sit on the topography or in the gaps in between. And so what happens then is it will resist the attachment of bacteria. But once they attach, there is no mechanism to try to essentially disrupt the bacterial cell wall or kill them. In contrast to that, in about 12, 14 years ago, there was a paper where the first reported study from a group in Australia, where they noticed that insect wings, in particular for a cicada, and certain subspecies of cicada, the insect wings have these nanoscale features where the feature size is much smaller than the bacterial cell. And you can see here in the top right that the bacterial cell can essentially get penetrated by these nanopillars that are present on the insect wing. So this was the first such study, and what they showed is that even if you sputter coat it with gold, which means that you change the chemistry altogether, even then the bacterial cell would get killed. And so this was more of a what is now referred to as a mechanobactericidal effect, where you kill bacteria and not just resist them. And that was maybe the start of a subfield or an effort to try and find many such surfaces or other surfaces in nature. And since then, there have been studies where people have found that there are very high aspect ratio structures on dragonfly. And also, then on the bottom right, there's a gecko skin. So many of these have these nanoscale features. There are subtle differences in terms of their aspect ratios and also how they're ordered. The one on gecko has a more tent-like arrangement. The one in the dragonfly would have a more a disordered structure versus the one in the cicada has a more ordered structure where and also the heights would differ, but they more or less work in a similar fashion in terms of killing bacteria through mechanical contact. So, Anindo, next one. So now with that report or these studies, people have tried to see how to replicate them on surfaces that would be of use for a variety of applications. The first few studies were on silicon wafers, and these are some of the efforts from our own group, but there are, of course, many other groups in the world who've been trying to do that. So what you see on the left panel are basically these structures that we've created on silicon wafers by reactive ion etching. And probably the field started off with silicon because of the semiconductor industry that's been using these technologies to etch silicon and generate these kind of high aspect ratio structures. So you can see here in the higher magnification SEM image that these are tall pillars that have been generated on silicon wafers. And what the bottom image shows are bacterial cells that are sitting on it. You can probably see they're kind of stretched out, and that's because they've adhered to the pillars and sort of they try to make contact, they stretch out, and they're like torn apart or ripped apart, and that's how they get killed. Uh, you can hopefully make out some red dots, and that is because we've added a dye. In these experimental results, if we add a dye, if the cell is dead, it will appear red in the fluorescence microscope. If they're alive, they will appear green. So what these says, I mean, these images are saying that cells are dead when they come in contact with the surface. Similarly, in the middle one, we switched over to titanium. So this is commercially pure titanium. So you can see here in the top image, there's a shiny um, coupons of titanium that we have placed on the silicon wafer. We put it for reactive ion etching. After 10 minutes, when it comes out, it has these nanopillars, and which gives them the black appearance because light doesn't reflect so well from these surfaces. And here again, if you see the bottom panel there, the bacterial cells are essentially ripped apart, and they are mostly red in color because they are dead and they take up the red dye. So these techniques use reactive ion etching, the ones that are used commercially, or I would say that are used for semiconductor industry. Uh, People are now looking at softer substrates like plastics or even gels. And what you see on the right panel are essentially these structures generated on PET. And again, this is Anindo's PhD work. And what you see here are, these pillars that form higher ordered structures of like a tent-like arrangement. And then again, when the bacterial cells sit on it, they make contact, but they do get ripped apart, and finally a large fraction of them do die. So, but depending on what the arrangement is, what the aspect ratios are, you get different fractions of death. So the amount of red versus green cells will depend on the kind of arrangements we have on the surface. Anindo, the next one. So in trying to understand this, there have been a lot of modeling that people have been trying to do. And of course, there are conflicting theories on how these things are sort of are probably working, because it's very difficult to experimentally visualize them. So people have come up with thermodynamic models and also other FEM-based models. And at this point, I'll hand it over to Anindo. He can explain to you what some of these current models are, their limitations, and of course, how he's been trying to address them through some, developing newer models. Anindo, all yours.
Yeah. Uh, Thank you for the introduction, Professor Prakash, and thank you, Kaushik sir, for the excellent introduction for the first few slides. So, as Kaushik sir explained, the one of the key goals is to emulate these surfaces on relevant substrates so that we can then translate them in for practical applications. And to do that effectively, we need to understand how the dimensions of the nanostructures affect the way in which bacteria interact with them. And that would help us decide which surfaces or which nanostructures would be more effective in eliminating the bacteria compared to the other ones. To this end, people have tried out different approaches. For example, I'll start with the thermodynamic models. So here, basically, the idea is that you have a bacterial cell, as you can see here on top of the nanostructures. And then this is the initial stage when the bacteria is just coming and resting on the top of the nanostructures, just reaching the surface. So this is our initial state. And in the final state, the bacteria will get pierced and ruptured. So we want to see if this process is thermodynamically favorable, and then try to calculate what sort of dimensions would be good for this process to occur more efficiently, and also try to understand how more nitty-gritties of the process, as in where the rupture is occurring, how it is occurring, and so on. So to this end, as you can see, we can calculate the change in free energy between these two states uh, using the idea that when the bacteria comes and sits on top of the nanopillars, there will be a tendency of the bacterial cell wall in the vicinity of the nanopillar surface that will try to attach to the cell wall will try to absorb onto the nanopillar surface, because bacteria by their nature, they seek and attach to surfaces. So, but while this process is happening, it will also wrap around the nanopillar, and that will stretch the cell wall, and the cell wall also needs to bend around the nanopillar. So there will be a stretching, a penalty for the stretching energy and the bending energy, whereas the adsorption process uh, is so these stretching and bending processes must be compensated by the decrease in free energy that is afforded by the adsorption of the bacterial cell wall onto the nanopillar surface. So then using this equation, we can then define geometries and then formulate equations, and then turn this into an optimization problem. Uh, and then based on, so for example, here I picked one example from one of the more popular studies, where they have taken a hemispherical cone with or a paraboloid geometry for the nanopillar. And we can assume that the bacterial cell wall, so the bacterial cell wall is actually very complex with multiple layers. But for this purpose, we can assume that the bacterial cell wall is a thin single layer, because usually it is of the order of 10 to 20 nanometers, whereas our pillars as well as the bacterial cell itself are at least one order of magnitude higher in dimensions. So, and then by formulating equations and then establishing constraints, which is usually that the projected area for the initial state and the final state should be constant, we can then calculate the stretching degree in the different parts. In the part where the cell wall is adsorbed onto the nanopillar itself, and also in the part where it is suspended between the two nanopillars. And if we do that, we'll see that the stretching degrees, they increase as so we can plot them against something called the effective interaction parameter, which is basically the ratio of attraction of the cell wall to the nanopillar surface, or how aggressively they will adsorb onto the nanopillar surface, to the ratio of cell wall elasticity, or how rigid or robust your cell wall is, how much it can stretch before it undergoes rupture. So by doing this, the main finding that we can reach is that the rupture that occurs is actually occurring in the region suspended between the two pillars. It's not occurring at the pillar tip or the region which is adsorbed on the pillar. So the critical action site, we can gain insights about the critical action site. And also, since we are defining these stretching degree, the bending energy, as well as the adsorption free energy in terms of the bacterial geometry and the nanostructure geometry, we can then use those equations to plot the stretching degree against the pillar radius and interspacing, or basically the parameters that we use to characterize the nanostructured surface. If we do that, what we will see is that, and then we can plot sort of this sort of phase maps. And as we can see here, at very low pillar radiuses, there will be a suppression phase. Now, this occurs because as your pillar radius decreases more and more, beyond a certain level, the bending energy reaches very high values. So it becomes thermodynamically impossible for the adsorption-free energy to overcome that bending-free energy, and therefore, at such low radius values, it is very unlikely that the cell wall will actually wrap around the nanostructures. So therefore, at very low pillar radiuses, thermodynamically, the process seems unfavorable. And similarly, if you increase the pillar radius, the bending free energy decreases, so you have more and more stretching degree. Now, with respect to interspacing, at very low interspacings, the surface is not very good for mechanobacterial activity, because it essentially acts as the interspacing keeps on decreasing. As it reaches zero, it essentially acts as a flat surface. So we need some sort of interspacing, some sort of features spacing between the features, so that it can penetrate the bacterial cell. But also, if you keep on increasing your interspacing, then again the bacteria can settle in between your nanostructures, and then again it is not very good for the bactericidal efficiency of the nanostructured surface. So what we want is a relatively high pillar radius with a relatively low interspacing. And the two dots you see here, these are the pillar radiuses and interspacings that are noticed generally for the cicada wings, different species of cicadas. And as you can see, they are in the enhancement phase, which explains why the cicada wing has such a good mechanobacterial activity. Now, the thermodynamic models are for understanding the overability of the process. It doesn't actually tell us anything about how the process is happening. So what are the driving forces that are responsible, and so on. So, and also it doesn't allow us to understand the stress distributions and other things. So therefore, the finite element method is a good alternative approach to understand these things. So here, as you can see, again, we can simulate. So basically, if in case you are unaware of the finite element method, it is basically a method to calculate stresses and strains for complex geometries through a deformation process as the process progresses. It is a computational method usually based on the Newton-Raphson method. So what we generally do is we take a body with a complex geometry, and then we divide the body into smaller parts by meshing it, and then calculate whatever quantities we desire at each mesh point, and then extrapolate these values between mesh points to generate stress fields, as you see here. So again, in this case, as you can see, this is the bacteria. And one of the advantages with the finite element method is that you can apply symmetry conditions. So for example, here, assuming that we have an ordered nanopillared structure, we don't need to simulate the whole bacterial cell. We can just take a small area as shown here. And then even here, we don't need to simulate all 4 pillars or even one complete pillar. You can take 4 points, which are denoted as L1, L2, L3, and L4, and then simulate them like this. So you only simulate 1/4 of the pillar and 1/4 of the area between 4 pillars. And the bacterial cell wall we can simulate as 3 layers, which is more common, which is actually common for the gram-negative bacteria. And for the other strains of bacteria, which are gram-positive strains, there are only 2 layers. But through experimental investigations, it has been found that gram-negative bacteria are generally more susceptible to the mechanobactericidal effect, which is why in this study, the authors focused on the gram-negative bacteria. So there are 3 layers in the bacterial cell wall. So instead, unlike the thermodynamic approach, as you can see here we actually resolve the cell wall in order to understand the stress distributions in each layer. And that can provide us more insightful understanding of the mechanobactericidal process. So the lower layer is the outer layer. Then you have the cell wall in the middle, and then you have the inner layer. So the outer layer and the cell wall, these are covalently linked with lipoprotein. So the stress transfer from the outer layer to the cell wall is actually very efficient. But then between the cell wall and the inner membrane, we have a fluid-like layer of something what is called periplasmic space. So the inner layer and the cell wall, they can actually slide against each other, albeit not very much, but they can slide against each other. And stress transfer between these 2 layers is not very efficient. But the outer layer and the cell wall, they are covalently linked. So then to simulate the adsorption process, what we can do is we can apply a downward load on the outer layer, which is the bottom layer here, and then progressively increase that load in subsequent steps to simulate the progression of the adsorption process as the bacteria adsorbs more and more onto the nanopolar surface. If we do that, and then we look at the circumferential uniaxial strain and longitudinal uniaxial strain in the cell wall, the inner leaflet and the outer layer, we can find out the distributions in these layers. Now, from these distributions, we can also calculate the aerial strains, and then look at the 4 different points that we have defined and see how these strains are distributed in these 4 points. And that can tell us which layer is strained most at which point, and therefore help us understand where the rupture is actually occurring, why is it occurring, and how is it occurring. So as you can see here in the outer leaflet, the L4 or the point suspended in the suspended region between the pillars has the maximum aerial strain. So this x-axis is something of a dimensionless parameter called sinking depth. So as your sinking depth is increasing, the adsorption is progressing slowly more and more. So basically the pillar is penetrating the bacteria more and more. So as that occurs, as you can see, the aerial strain in the outer leaflet increases much more significantly at L4 point compared to L1, L2, or L3, which tells us that in the outermost bacterial layer, cell wall layer, the maximum aerial strain occurs at L4. But if you look at the inner leaflet, the inner layer, you will see that the maximum aerial strain occurs at L1, which is at the pillar apex. So which you can also see from this simulation here. So this tells us that it is not necessary that across all the different layers of the bacterial cell wall, the stress distribution will be uniform across all the different points. And yeah. So if we compare the maximum aerial strain for the inner leaflet and the outer leaflet, we will notice that this maximum aerial strain in the inner leaflet is much more higher at comparable sinking depths than the outer leaflet. So therefore, if rupture is occurring, it is more likely to occur at point L1 because at similar sinking depths, that point has much more aerial strain compared to the outer leaflet. And also if we compare the maximum so not the aerial strain, but the uniaxial strain, again, we will notice that at L1, it has the maximum uniaxial strain as well. So contrary to the predictions of the thermodynamic model, a finite element modeling with a more resolved view of the bacterial cell wall tells us that the critical site of the rupture or critical action site actually occurs at the pillar apex and not at the suspended region between the pillars, even though for the outermost layer, the suspended region experiences the maximum strain. We can also we can also investigate the role of the nanoscale geometry. So again, the role of pillar radius and the role of interspacing. So if we look at the role of pillar radius, you can see that similar to the predictions of the thermodynamic model, as the pillar radius decreases, you will have significant increase in the maximum strain, and as pillar radius increases, the areal strain will decrease. However, unlike the thermodynamic model, in the finite element model, there is no limitation for how high the maximum strain can go when you keep on decreasing your pillar radius. So therefore, we have to be really careful with the interpretation below 20 or 10 nanometers of pillar radius when we are using the finite element model. In case of interspacing, as you can see, both for the areal strains and the uniaxial strains, there is not much difference even when we increase the interspacing from 120 to 180 to 240 nanometers. So as you can see, they almost overlap. Only at very high work of addition. Work of addition is basically the energy that will be released when the adsorption occurs, because that is a thermodynamically favored process. So when the work of addition is very high, that is the bacteria is very aggressively likely to adsorb onto the pillar surface. It likes the nanopillar surface very much. It would like to bind to it. In such cases, at very high work of additions and the areal strain can begin to deviate slightly with changes in interspacing, but even then these differences are less than 5%. So what the finite element model tells us is that interspacing has a much lower role in the rupture process compared to pillar radius. The pillar radius is actually the more dominant parameter. Now, so till now, we have assumed that the adsorption is the only force that is responsible for the wall rupture. However, recent studies have shown that there can be other external forces that are responsible. So, for example, here, what the authors have done is that they have taken a flat silicon substrate and a nanostructured silicon substrate, and then they put a droplet of bacterial suspension on top of the substrate, and then introduce a air bubble into the droplet. So what that does is that will allow the air bubble to transverse through the droplet, and therefore there is an air-liquid interface, or ALI, that is passing through the bacterial cells that have adhered on the surface. And what they noticed is that in case of your flat surface, that doesn't affect the bacterial cell very much. But in case of your nanostructured surfaces, when the air-liquid interface passes, soon afterwards, the number of almost all of the bacterial cells are dead. And this can occur as quickly as 120 seconds or 2 minutes. So to understand this, what is actually happening here is that when the air-liquid interface passes through the surface, through the substrate surface and through the bacterial cells, they will exert downward capillary forces because of the meniscus, liquid meniscus that passes through the surface. And that will generate a downward push on the bacteria, which can actually push the bacterial cells onto the nanostructures, causing them to rupture. And therefore, this can enhance the mechanobactericidal activity. And in case of certain surfaces where they inherently do not have very good mechanobactericidal activity, they can impart them significant mechanobactericidal activity. And we can we can calculate this downward component of this force using this equation. So basically, we can model the bacteria as a hemisphere, a cylinder with two hemispherical ends. So this way, we can model either rod-shaped bacteria, like I described, or we can also model spherical bacteria by taking the cylinder length to be 0, and then the two hemispherical ends come together to become a sphere. And there are certain assumptions. So, for example, we assume that the contact angle of the bacterial cell wall with the liquid is constant and it does not change. And also, when this sort of meniscus will pass through the bacterial cell, there will be pressure differentials, which we assume to be negligible and very small or negligible. With these assumptions and using this model and this equation, we can calculate the maximum peak force that a bacterial cell would experience. So, for example, P. originosa or Pseudomonas aeruginosa is a very well-known rod-shaped bacteria, and it is actually responsible for lots of bacterial infections, hospital-acquired infections, and even implant infections every year. So we can calculate the maximum peak force. We will find that the maximum peak force experienced by the bacterial cell would be in the range of around 250 nanonewtons. Whereas for a spherical cell, for example, S. aureus or Staphylococcus aureus, again, a very well-known opportunistic pathogen, the maximum peak force only reaches around 100 nanonewton. So not only the nanostructure geometry, not only the nanostructure dimensions, nanostructure features, but even the shape of the bacteria also has an effect on the maximum peak force it can experience, and therefore how likely it is to get killed via external capillary forces. So now, up till now, we have focused on mostly on the bacteria, and there is an assumption that the nanostructure themselves they do not bend or deflect under these sort of external forces or under forces that are exerted by the bacterial cell on the nanostructures after they attach to the pillar tip. However, that is not always true. As you can see here, when the aspect ratio of the structures increases. So, for example, if you fabricate pillars with comparable diameters or same diameters but increasing height, so therefore increasing aspect ratio, what we can notice is that for low aspect ratio structures, the pillar, they maintain their original morphology. They are perfectly erect, rigid, and ordered, comparable to their initial state. However, as the aspect ratio increases at higher and higher aspect ratios, the bacteria can exert forces on the pillar tips and cause them to bend and deflect. Now, when this happens, using Euler's theory of buckling, we can calculate the maximum deflection that it will experience based on the force that is exerted by the bacteria. And then what this process does is actually it will store strain energy when the pillar is being bent. And obviously there is a restoring force as well. Now, when this stored strain energy crosses a certain threshold value, the pillar will spring back to its original position and it will rupture the bacterial cell wall. So as you can see here in these two images. So this is actually another mechanism, another way in which higher aspect ratio nanostructures, flexible nanostructures, can kill bacteria. Another important point to note here is that depending on the aspect ratio, for same pillar diameter, if we increase the length, so increase the aspect ratio, for same amount of tip deflection, the stored elastic energy in a taller pillar will be much lower than that of in a shorter pillar. So therefore, basically, shorter pillars are more difficult to deflect. And when they do deflect, they store much more strain energy at similar tip deflections compared to their taller counterparts. And therefore, for similar pillar diameters and different pillar lengths, and if we culture bacteria on top of them, shorter, higher aspect ratio pillars are more likely to kill higher amount of bacteria compared to much taller pillars. So just increasing the length infinitely will not keep on enhancing the mechanobactericidal activity. So with these things in mind, we designed our own study. What we did was we fabricated higher and higher aspect ratio nanostructures on titanium using reactive ionizing, similar to the structures you saw in a previous slide. What we did was, in order to expose them to the capillary forces, so we culture the bacterial cells on top of the surface and then incubate them for a certain time, which allows them to go and adhere to the pillar tips, to the pillar substrate. And then we gently wash the substrate surface to remove any cells that have not adhered to the substrate or are loosely adhered. And then we can process these through two different routes. In one case, we allow the sample surface to dry very briefly for 4 to 5 seconds, which allows the air-liquid interface to pass through the substrate. And in other case, we keep the substrate dry throughout, and then we can add the fluorescent dye, combination of green and red dye, and then image them while the substrate surface is wet. So if we do this, what we noticed was that in both, so this is in case of the wet condition where the sample substrate is wet throughout, and this is in case of the dry condition where they have been allowed to dry very briefly. So there are two key observations. First observation is that as the aspect ratio of the nanostructures increases, less and less bacteria die, as is expected. And also, the other important observation is that when the sample substrate is allowed to dry very briefly, the killing efficiencies for all the surfaces get enhanced significantly. Although the effect of aspect ratio is still maintained. Now, although these are high aspect ratio structures, they are not extremely high aspect ratio so as to become very unstable or not to be rigid enough to be able to penetrate the bacterial cell wall. So we wanted to see if there are any other effects responsible for the low killing activity of the high aspect relatively high aspect ratio structures, even after the external capillary forces are allowed to act. So to do that, we imaged the cross section using FIB. So this is for the T1 surface, which has an aspect ratio of 34.83. This is for the T2 surface, which has an aspect ratio of around 88. And this is for the T3 surface, which has an aspect ratio of 120. So you can see very similar to what we observe in insect wings. The bacterial cell has been penetrated and ruptured. And if we look at the cross section, we can see the cell has sunken between the nanopillars and the intracellular contents have been leaked out. The cellular morphology is not maintained at all. Similarly, for T2, part of the bacterial cell has been ruptured and a small part is still yet to be ruptured. But if you look at the cross section, the pillars are all erect and rigid, and they have penetrated the bacterial cell wall similar to T1. However, if we look at T3, we will notice that the pillars, they have buckled, they are bent, they are not rigid and erect, comparable to their original morphology, which you can see here in the background, which tells us that when the capillary forces are acting under these forces, when the bacteria is pushing down on the nanostructures, if your aspect ratio is below a certain critical value, the nanostructures, instead of penetrating the bacterial cell wall, they are more susceptible to buckling failure. And when that occurs, they won't be able to penetrate the bacterial cell wall, and therefore they won't kill the bacteria, which is the reason for the decrease in mechanobactericidal efficiency with increase in aspect ratio that we notice here. So then, now, if you remember from these equations, apart from the applied load and the dimensions of the nanostructures, which are expressed through the parameters P and L, we also have the elastic modulus or the substrate property. So that also affects the nanopillar bending ability and how rigid they are and so on. So therefore, we wanted to test on softer substrates, what role does how does the mechanobactericidal activity vary with aspect ratio in case of softer substrates compared to silicon or titanium, which have a much higher elastic modulus in the range of GPa. The bacterial cell wall has elastic modulus in the range of kPa. So whereas this softer substrates, they will have elastic modulus in the range of MPa. So we fabricated two different nanotopographies on PET. So PET, as you know, is a very well-known polymer. It is used extensively, starting from water bottles to all sorts of touch surfaces for your phone screen guards and so on. So we prepared one surface to mimic the cicada wing with a relatively low aspect ratio of 4.34, which we will call the nanopillared surface, and another one with a much taller aspect ratio with a height of 500 nanometers and diameter of only 25 to 30 nanometers. So this is more mimetic of what we find on the dragonfly wing surface. So these are the physiochemical characterizations. As you can see, because these were generated through plasma treatment, this renders them super hydrophilic. And apart from that, there is not much change in the bulk of the substrate because plasma treatment is a surface treatment, so it does not affect the bulk surface. So once again, we tested them for with capillary forces and without external capillary forces. And as you can see, for the nanopillared surface or the low aspect ratio surface, usually the killing activity is much higher compared to the nanowires or the higher aspect ratio ones. But when external forces are allowed to act, in this case, we observed that in both cases the killing activity reached almost 100%, more than 90%. So this was puzzling because for the nanowire surface in the dry state, the killing activity should be still be lower than the nanopillar surface due to its higher aspect ratio. So when we looked at SEM images, we noticed that there are basically two sorts of interactions. In one case, the pillar are basically bacteria. They can either undergo compressive loading or they can undergo buckling failure depending on their aspect ratio and other things. Whereas in case of the pillars that are adhered to the tip of the periphery of the bacterial cell, they will undergo a shear deformation, a shear force at the pillar tip. More closer observation, through more closer observation, we discovered that in case of the higher aspect ratio nanotopography, actually the pillars no longer remain as individual erect pillars, but they form the stent-like superstructures due to interpillar tip interactions. Now, through FEM, we calculated the loading conditions. So for the nanopillar surfaces, obviously they only undergo compressive loading. And if you look at the maximum stress that they experience, this is only in the range of kPa, uh, whereas the maximum strain or stress that PET can under the yield stress for PET is in the range of 50 to 70 MPa. So therefore, it's much higher. And these pillars, they act as rigid bodies. They do not deflect or bend or fail under these conditions. However, for the T2 or the nanowire surfaces, if we take individual single pillars and we calculated the buckling load through both FEM and also through Euler's theory for buckling of linear columns, we find that the loads that we calculate are in the range of 1 to 3 nanonewtons, which is actually comparable to the load a single nanopillar would experience under the action of the capillary forces when the air-liquid interface is passing. So therefore, if these structures remained as single pillars, then maybe we would have noticed a lower killing activity in case of the nanowire surface under the dry condition. However, what is happening here is that due to the interpillar tip interactions, these pillars, they come together and form tent-like structures, which imparts them with enhanced rigidity. And that allows them to actually penetrate the bacterial cell wall, even though single pillars would fail under buckling. We also simulated their behavior under shear forces for both the topographies. So, and if we plot the force versus displacement curves for these two topographies, we will notice that their behavior under forces experienced by a single pillar, which would be obviously lower than 20 nanonewtons, are actually comparable. They are actually very similar. So therefore, for softer substrates or for certain substrates, the interpillar tip interactions can give rise to formation of superstructures, which can actually modify their behavior and impart them with enhanced rigidity and therefore increase their mechanobactericidal efficiency. So it's not just the properties or the role of these individual pillars, but we also need to consider consider interpillar interactions and other effects. That's all. Thank you. In case if you please ask if you have any questions.
So thank you, Professor Kaushik, and then Dr. Anindo. We had an excellent talk, and I'm sure there are some questions from the audience. If you would like to type in the questions in the chat box, we can take it up one after another.
Can I just speak?
Sure. Yeah, those who can speak,
So
yes.
thank you, Anindo. This it's very interesting. I just wanted to understand this distinction between the thermodynamic argument and the FEM with respect to why very narrow or very small radii of curvature in the one case should not even allow adsorption, whereas in the other case it seems to uh, not consider it at all. What is the difference? I mean, the FEM also includes the same interactions and forces that you have in the thermodynamic argument. So why doesn't it predict that a very narrow tip will not lead to the bacterium wrapping itself around?
Right. Yeah. So, sir, actually, the interactions are similar, but the way the interactions are then calculated or that is different. So, for example, in case of your finite element model, as you can see here, what we are basically doing is we will apply a downward force with increasing radius on the lower layer. So let's say you have a pillar with diameter 50 nanometers and you have another pillar with diameter 10 nanometers. So as you and we are applying similar loads in both cases. So when you have the pillar with the lower diameter, it just acts as a more penetrative surface, and that is all there is to it. There is so it's strictly
Sorry, can I just interrupt for a moment? When you say apply more loads, you're just allowing the surface forces to act, right? You're not applying anything. This is like
no.
JKR or something like that.
No. So yeah, I'm sorry, I should have explained that part. So here in FEM softwares, there is actually no way uh, to simulate the surface adsorption as it occurs in nanopillar and bacteria. That is formation of bonds and so on. So therefore, what we do is we will progressively apply just mechanical load or downward force on the lower layer. And then once the points come in contact, we will put friction conditions such that there is no slip. So it's essentially mimicking the adsorption process, but it's not actually the formation of bonds or surface forces that would actually occur in real life. And whereas in case of
Okay, there is a question from Haritra Chatterjee. Would you read it on the chat box, or you want me to read, Anindo?
no, sure, sir.
He's talking about the critical number of wires or beams required for the formation of the superstructures.
Right. So there is no critical number as such. It is just that if your pillar density is below a certain, so if your pillar interspacing is not too high, as such, it allows the pillars to interact with each other through electrostatic interactions or capillary forces or van der Waals forces at the pillar tip. And your pillars are not spaced too far, then they will come together to form superstructures. Now, the exact number of pillars that will come together, that depends. Even in the same nanotopography, there can be superstructures with, let's say, 4 or 5 pillars coming together, and there can be superstructures with more number of pillars, maybe 10 or 12 coming together. We chose 5 pillars because, as you can see in the SEM image here, that was, so if you see here, roughly there were 4. So I don't know if I can zoom this. Yes. So if you see, it's 1, 2, 3, 4, 5. So 1, 2, 3, 4, 5. So it was for us, for all the tips that are coming together, it was roughly around that value. Based on that, we chose this. But Yeah, it can be more than that. Sorry.
Okay. Are there any other questions from the audience?
Actually,
Yeah, I had one more.
Sure,
Yeah.
please go ahead.
So I don't see anything in the chat box. The this slide 6 where you show the fact that the thermodynamic argument predicts rupture in the flat portion which is between two pillars. So there must be some tension acting there, which is a result of some surface forces on the curved part of the pillars.
Yes, sir.
Are they
So
stretching or something, or what's going on here?
yeah, basically there will be stretching in both the regions. The regions that is the region that is absorbing on the pillar surface, as well as the region that is suspended between the pillars. So the way this works is that you have when initially the bacteria is coming and attaching on top of the pillar, and then the point immediately next to the pillar surface that will try and get absorbed on the pillar surface. And progressively, as this happens, uh, what will essentially happen is that this area will stretch to accommodate the increase in the in the curvature, or increase in the total area here. So basically, it's your suspended region is basically getting stretched as more and more pillars, because this point at the pillar tip, that is the initial point of contact. So as soon as the bacteria comes and rests on top of the pillars, that point is attached, and that cannot move now. So whatever increase in area you are having because of the cell wall wrapping around the nanopillar, that has to come from the region suspended between the pillars. Did I answer your question?
Yeah. So the stresses are the same everywhere, but the suspended part is not attached to anything, so it can break more easily than having a rupture in the hemispherical part. Is it?
Uh, no. The so 1 minute. So you see, this point initially the bacteria attaches here, and then
Right.
let's say right now at that point the SB region will be a straight line here. So if you compare the perimeter or length for that region compared to this complete region, this is obviously much more. So, but that increase, how is it getting accommodated? Because this point is now fixed. This point initially, when the bacteria comes and attaches, that point becomes fixed. It cannot now move around. So therefore, let's say this was 1 unit, and after stretching, this has now become 1.5 units. So that 0.5 unit increase has to come from the region that was initially suspended between the pillars. So basically, as the adsorption progresses, more and more points, as they will get absorbed, they essentially become immobile. They are fixed on the pillar point there. So now only the region that is
the
suspended.
strain doesn't build up on the curved surfaces.
There will be certain small amount of strain on the curved surface as well.
Yeah.
But mostly on the region suspended. Yeah.
Yeah. Anindo, there's actually a question from Dr. Deepa Srinivasan. She messaged me because she's traveling. So she's asking whether, based on all of these calculations, would it be better to have a random distribution, like say like a bimodal distribution of pillar features? Would that be optimal, or I mean, what's your thought on that?
So actually, there is not much. There have been studies that have looked at the effect of order and disorder in the nanostructure topography. There is not much difference between these two, simply because biological processes, they by themselves are stochastic in nature. So therefore, if you have a ordered structure or a disordered structure, is not going to affect the individual cells that much, because the cells themselves, they will have a more random distribution of their behavior. So yeah, there is no particular advantage of having a strictly ordered topography, other than if you want to investigate the role of exact nanostructure dimensions, let's say radius, then you want to increase or decrease or control the nanostructure feature dimensions precisely. Other way to look at it would be when you want to translate this technology, you want to manufacture it on complex geometries on large surfaces. You would ideally want processes that can that can do that sort of thing. And those such processes, bottom-up processes, they generally produce a more sort of disordered structures, not exactly ordered structures like lithography or photolithography. I think there is one more question in the chat box.
It talks about RIE
Yes.
byproducts
Yeah.
interaction.
So RIE is basically a subtractive process. So therefore, whatever nanostructures you see here, what essentially happens is, let's say in case of SiO2, initially your silicon wafer will have a layer of SiO2, very thin layer of SiO2. And when the process starts, that will get physically sputtered away. Similarly, in case of titanium as well, it will initially have a thin layer of TiO2. And when the sputtering process starts, it will first get physically sputtered away. That exposes the underlying commercially pure titanium or pure silicon that can then get etched through both chemical processes and physical sputtering. So because the etching progresses in the bulk pure substrate, there is a very low possibility of any external contamination during the etching process. And essentially, whatever nanostructures you are generating through this process, these are the same material as your bulk substrate. So in this case, it will be pure silicon, and in this case, it will be pure titanium. Now, obviously, when you take them out from the RIE chamber and expose them to atmospheric oxygen, there will be a thin layer of oxide that forms, but that is no different from the flat substrate. So therefore, there is no difference. And as Kaushik sir explained earlier, even in case of naturally occurring surfaces, to verify if their chemical composition has any effect on this bactericidal activity, what people have done is that they have coated the surfaces with a very thin layer of gold, 10 to 20 nanometers, and then compared them with uncoated surfaces, and they did not notice any difference. So therefore, this effect does not arise because of the chemical composition or the chemical components of the surface itself, but because of the physical interaction of the bacterial cell with the nanostructures.
Anindo, I think I have some
Yeah, sure.
clarifications to see. See, when you talked about in the slide 6, the pillars, initially I thought it is a rigid pillar, but I think later you mentioned that it is a flexible pillar that you have considered, right?
So, sir, for all the thermodynamic models, essentially everybody considers the pillars to be rigid. Only more recently, our group and a few other groups, we have started experimenting with much higher aspect ratio structures when the pillar becomes more flexible, and then all of these aspect ratio effects come in. But if you look at the naturally occurring structures, for example, on cicada wing or even on the dragonfly wing or gecko skin, or if you are mimicking them on very rigid ceramic substrates, for example, silicon, for all purposes, we can assume the pillars to be rigid.
Okay. Do you allow the thinning of the bacterial wall during your simulation?
So, sir, for the
See,
finite
since
for
you
this
have
one,
mentioned here in
yeah.
your finite element, if you go back to slide 6, where you said that at SB there is a what do you call this, sticking or adhesion, and then later on you are expecting that it will take the shape of the pillar and then there will be a flat surface. There are, there is a possibility if there is a friction that is taking place between that bacterial cell wall and then the pillar, that can change the shape in which this configuration is taking place, isn't it?
No, sir. Actually, that is not possible, because what essentially happens is that when the bacterial cell will land on top of the pillar, as the cell wall progressively wraps around, it will form molecular bonds with the substrate. So therefore,
Okay.
essentially, it's they do once one a point on the bacterial cell wall adsorbs to a point on the nanopillar surface or even on a flat surface on a point on the surface. it does not then move around. So then it is actually fixed at that point. Yeah.
Okay, then what about the wall thinning? Why does the rupture take place? Is it because of the wall thinning?
Yeah. So basically, as your this sorry, as your cell wall is progressively wrapping around the nanopillar more and more, the suspended region between the two pillars that has to stretch in order to accommodate this increase in the perimeter.
Right.
And beyond a certain point, if you keep stretching a thin sheet, thin membrane, if you keep on stretching, beyond a certain point, it will rupture. So that is what is happening actually.
Another point that I wanted to get clarified is, see, you are using finite element method.
Yeah.
For the analysis, right? But then, you have to also feed in the material constants.
Yes.
So where do you get those material constants?
Yes. So so these are generally taken from different studies that have been conducted over the past, I think, 2 or 3 decades. So for bacterial cell wall, the assumptions are there that the material is neo-Hookean viscoelastic. And the properties, they are generally not isotropic. So orthotropic assumption is the most common, but people have also assumed them to be isotropic. To answer your question, where do we get the exact parameter values from or the parameter constants from? So, for example, you can take a single bacterial cell and then take a capillary tube and then create suction on the cell wall surface, and from that you can calculate various material properties. Similarly, you can take an AFM tip, poke a bacterial cell that has attached to a surface, poke that bacterial cell with it, and then from the force displacement curves, via some analysis, you can calculate different material properties. And so these studies have been done previously in the 2000s, in the 2010s. We use those parameters.
Good. Yeah. So if there are no more questions from the audience, then I would like to thank Professor Kaushik Chatterjee and Dr. Anindo Roy for this excellent, interesting talk on this, the nanomechanics of bacterial cell wall damage via the high aspect ratio bioinspired structure. And I thank the speakers once again. Thank you. And next month, I think we will have the webinar by Dr. M.D. Matthew, formerly with the Indira Gandhi Center for Atomic Research. He would talk on the creep and related aspects of structural integrity. So with this, I will now thank once again the speakers and then the audience for having attended this webinar. And we will kind of complete today's webinar. Thank you very much. Thanks for joining.